Steps in cleaning validation, with examples. Presented by Learn GMP Incorporated in collaboration with Technical Training and Consultation Service LLC. Introduction. Cleaning validation is a critical process in pharmaceutical manufacturing to ensure that residues, from actives, excipients, and cleaning agents, are effectively removed between product batches or after maintenance. Here's a breakdown of key steps with examples. The first step is to create a Cleaning Validation Master Plan, CVMP. What it includes: scope and objectives, equipment list, cleaning procedures, acceptance criteria, for example, MACO, responsibilities, sampling and testing strategy. Example: the CVMP may state that all tablet coating pans will be validated using swab and rinse sampling with a PDE-based MACO limit. The next step to perform the risk assessment and worst-case identification. How to do it: evaluate products based on toxicity, PDE, solubility, batch size, cleanability. Pick worst-case product and equipment train for validation. Example: drug X with low solubility and high ADE is selected as worst case for the granulation line. Define cleaning procedure is another main step, which includes manual versus automated, CIP, detergents used, parameters like time, temperature, rinse volume. Example: a manual cleaning process may involve 5-minute hot water rinse, detergent scrub, final rinse, and drying. The next step to establish acceptance criteria based on use of MACO values, microbial limits, and visual cleanliness. Example criteria: residue less than 1.5 milligrams per swab, based on PDE. Microbial count less than 10 CFU/swab. No visible residue. For the sampling plan, there are two main techniques: swab sampling, for direct contact surfaces, for example, tablet hoppers; rinse sampling, for hard-to-reach areas, like pipelines. Example: swab 4 points on a tablet press, feeder, punch, die table, and shoot. Analytical method validation is a critical step in cleaning validation, which ensure the method used for residue detection is specific to current API, sensitive, LOD/LOQ below MACO, accurate and precise. Example: HPLC is validated to detect drug A at 0.1 ppm with LOQ of 0.05 ppm. Perform cleaning validation runs. To execute the cleaning validation, usually 3 consecutive successful cleaning runs are required. Example: run 1 to 3. After manufacturing drug X, cleaning is performed and residues measured, all below limits. Document and report. Create a cleaning validation report that includes test results, deviations, conclusions. Example: final report confirms all residues from drug X are below 1.2 milligrams per swab versus MACO 1.5 milligrams per swab. Validation is successful. Revalidation and monitoring. Perform periodic revalidation, for example, annually or after equipment/product change. Trending and change control. Example: new detergent introduced, partial revalidation of cleaning process. Thanks for watching our channel. Feel free to reach us at contact@learngmp.com for any training and consultation requirements.